Methods of analysis for the detection of genetically modified organisms. Qualitative nucleic acid-based methods. Nucleic acid extraction. DNA extraction. The basic principle of DNA extraction consists of releasing the DNA present in the matrix, and further concurrently or subsequently, purifying the DNA from polymerase chain reaction inhibitors. Method selection is an experience-based choice of the user. DNA quantitation. Quantitation of extracted DNA could be useful for subsequent PCR analysis. It may be performed by either physical, for example measure of absorbance at a specific wavelength, chemical, physical, for example use of intercalating or binding agents able to emit fluorescence, enzymatic methods, for example bioluminescence detection, or by quantitative PCR. There are several methods available to quantify the DNA present in a solution. It is for the user to choose the most appropriate one to be applied, depending on the amount and quality of DNA to be quantified and, consequently, on the matrix from which the DNA has been extracted. Preparation of the test portion. The test portion shall be of sufficient size and shall contain a sufficient number of particles to be representative of the laboratory sample, for example 3000 particles at an LOD of 0.1%, to allow a statistically valid conclusion to be made. For practical or technical reasons, it is not recommended to exceed a size of 2 gram. The methods for DNA extraction in test portions from 200 mg to 500 mg, which are usually adequate for DNA-rich raw materials, for example ground grains, flour. However, for certain matrices containing very low amounts of degraded DNA, insufficient DNA suitable for analysis can be extracted. In these cases, the test portion may be increased. DNA extractions shall be carried out at least on 2 test portions. Samples. All operations for the preparation of test samples, for example grinding, homogenization, division, drying, shall be carried out. For liquid samples, shake the vessel containing the sample to improve the homogenization of the product. In the case of non-homogeneous products like raw oils, check that the sediments have been completely removed from the walls of the vessel. For solid matrices that cannot easily be suspended, the matrix shall be ground to reduce the particle size and/or facilitate the extractability of DNA. In such a case, attention shall be paid to the particle size. Final food products that are solid or paste and have high lipid contents are often not easy to grind to the desired particle size in a single step. Several procedures may therefore be added, such as lipid removal using hexane after intermediate grinding, freezing or freeze-drying before grinding. In order to facilitate the grinding of paste or viscous products, it is possible to apply one of the treatments to certain matrices, that are heating to a maximum temperature of 40 degrees Celsius, dissolving in an appropriate liquid such as water, freezing at a temperature below or equal to -20 degrees Celsius. Homogenize the whole laboratory sample. Sample the 2 test portions, taking into account possible dilutions or concentrations. During milling or grinding, precautions should be taken to ensure that the heating of the sample is kept to a minimum, since heating can have a negative impact on the quality of the extracted DNA. Milling or grinding techniques with a high risk of cross-contamination, such as the combined use of liquid nitrogen and mortar, shall be avoided as far as possible. As a rule of good practice, any dust-producing methodological step should be contained from all other analytical steps. If salts, spices, powdered sugars and/or other substances that could potentially interfere with the extraction or analytical method are present, appropriate purification steps should be considered according to the selected method. For example, in samples from composite matrices, the target matrix, for example the breading layer of fish sticks, can be isolated for DNA extraction. DNA extraction and purification. DNA extraction and purification. To obtain a good quality DNA, it is advisable to remove the polysaccharides, RNA, proteins, lipid fractions and salts. Polysaccharides, pectin, cellulose, hemicellulose, starch, thickeners, etc., using appropriate enzyme treatments, for example pectinase, cellulase, hemicellulase, amylase, or organic extraction. for example CTAB or chloroform. RNA and proteins using an appropriate treatment, such as enzymatic treatment by ribonuclease and proteinase, respectively. The lipid fractions using, for example, enzyme treatments, or solvents, for example N-hexane. Salts, for example from the extraction or lysis buffer, from the precipitation step, able to interfere with the subsequent analysis. For solid or dried samples, the volume of lysis or extraction buffer should be adapted to guarantee the DNA is dissolved. DNA purification can be performed by different means such as fractionated precipitation, using solvents like phenol, chloroform, ethanol, isopropanol, and by adsorption on solid matrices, anion exchange resin, silica or glass gel, diatomaceous earth, membranes, etc. Several DNA purification principles may be combined. If appropriate, extraction and purification can be performed within the same step. When using vacuum freeze dryers to dry the DNA pellets obtained after a precipitation step, the risk of cross-contamination should be considered. Resuspend the DNA in water or in a buffer solution that prevents DNA from degradation. The controls shall be used according to table, applicable to DNA-based methods. As per the table, environmental control is recommended for all the steps from homogenization, nucleic acid extraction, assessment of nucleic acid quality, amplification, and assessment of result of nucleic acid amplification. Positive extraction control is mandatory for all the steps from nucleic acid extraction, assessment of nucleic acid quality, amplification, assessment of result of nucleic acid amplification, interpretation, and test report. Positive DNA target control is mandatory for all the steps from amplification, assessment of result of nucleic acid amplification, interpretation, and test report. Negative DNA target control is recommended for all the steps from amplification, assessment of result of nucleic acid amplification, interpretation, and test report. Amplification reagent control is mandatory for all the steps from amplification, assessment of result of nucleic acid amplification, interpretation, and test report. PCR inhibition control is recommended but mandatory in certain cases for all the steps from amplification, assessment of result of nucleic acid amplification, interpretation, and test report. The use of environment controls will help the laboratory to identify sources of contamination at an early stage, and can even be used to identify in which work area the contamination is present. At least one extraction blank control shall be included each time DNA is extracted from one or more samples. The tube shall always be the last in each series. It may be appropriate to put one extraction blank on, for example, a rack of 8 tubes or a microplate of 96 wells for automated extraction. A positive extraction control should be included regularly, and always when a new batch of extraction reagents is used. This control will reveal if something is wrong with the reagents or the performance of the extraction protocol. The positive DNA target control demonstrates the ability of the nucleic acid amplification procedure to detect the nucleic acid representative of the GMO or target taxon. This condition can also be fulfilled by an appropriate positive extraction control. The negative DNA target control demonstrates the ability of the nucleic acid amplification procedure to avoid false positive amplification in the absence of the nucleic acid representative of the GMO or target taxon. The amplification reagent control demonstrates the absence of contaminating nucleic acid in the PCR reagent batches used. The amplification reagent control can be omitted when the extraction blank control is used. The PCR inhibition control may be used to demonstrate the absence of soluble inhibitors. This may also be demonstrated by serial dilutions of the template nucleic acid. However, some type of assessment of the effect of soluble inhibitors on the results of the analysis of the sample shall be made. A PCR inhibition control is mandatory if all PCR tests on the sample are negative and for matrices where the yield of amplifiable DNA is not known. Different type of methods for the preparation of PCR quality DNA extraction that are phenol, chloroform-based DNA extraction methods, polyvinyl pyrrolidone-based DNA extraction methods, CETAS-based DNA extraction methods, basic CTAB method, silica-based DNA extraction methods, and guanidinium chloroform-based DNA extraction methods. Preparation of PCR quality DNA using the CETAS-based DNA extraction methods, basic CTAB method. The method consists of a lysis step, thermal lysis in the presence of CTAB, followed by several extraction steps in order to remove contaminants, such as polysaccharides and proteins. Alpha amylase is added to the lysis buffer to digest the starches in case of amylaceous matrices. Treatment of samples with proteinase K is necessary in a variety of matrices to eliminate proteins. Also, treatment with ribonuclease is usually recommended for those matrices where RNA co-precipitation may disturb the subsequent analytical test. The salt concentration during the extraction steps is very important for the removal of the contaminants, since a CTAB nucleic acid precipitate will occur if the salt concentration drops below approximately half a mole per liter at room temperature and if the temperature drops below 16 degrees Celsius. By increasing the salt concentration, for example addition of sodium chloride, the removal of denatured proteins and polysaccharides complex to CTAB is achieved, while the nucleic acids are solubilized. Chloroform is used to further separate the nucleic acids from CTAB and polysaccharide and protein complexes. Finally, the nucleic acids are purified by isopropanol precipitation and washing with ethanol. Sample extraction. Weigh 200 mg to 300 mg of the test sample into a 2 ml tube. Add 1.5 ml of pre-warmed CTAB extraction buffer and mix. In some cases a higher amount of buffer may be required to suspend the matrix. Centrifuge for 10 minutes at approximately 12,000 G force. Transfer the supernatant to a new 2 ml tube, add 0.7 to 1 ml volume of chloroform and mix thoroughly. Centrifuge for 15 minutes at approximately 12,000 G force. Transfer the upper aqueous phase to a new 2 ml tube. Next process is CTAB precipitation. Add 2 volumes of the CTAB precipitation buffer to the tube containing aqueous solution. Incubate for 60 minutes at room temperature without agitation. Centrifuge for 15 minutes at 12,000 G force. Discard the supernatant. Dissolve the precipitated DNA by adding 350 microliters of NaCl solution. Add 350 microliters of chloroform and mix thoroughly. Centrifuge for 10 minutes at 12,000 G force. Transfer the aqueous phase into a new tube. CTAB precipitation is not necessary for all matrices, only for protein and polysaccharide-rich matrices. Alternatively, a solid phase purification of the DNA, for example by the use of spin columns, is possible assuming the results are equivalent. Next and final process is DNA precipitation. Add 0.6 volume of isopropanol to the tube contains the aqueous solution, mix smoothly by inverting the tube and keep the tube at room temperature for 20 minutes. Centrifuge for 15 minutes at 12,000 G force. Discard the supernatant. Add 500 microliters of ethanol solution to the tube and invert several times. This is the critical step ensuring the complete removal of CTAB. Centrifuge for 10 minutes at 12,000 G force. Discard the supernatant. Dry the DNA pellet and redissolve it into 100 microliters of water or an appropriate buffer, for example TE buffer. This is the DNA master stock. The detection of genetically modified organisms, qualitative nucleic acid-based methods. Qualitative analysis consists of specific detection of target nucleic acid sequences in the test samples. Each method shall specify the target sequence. A qualitative result shall clearly demonstrate the presence or absence of the genetic element under study, relative to appropriate controls, and within the detection limits of the analytical method used, and test portion analyzed. PCR amplification. Amplification of the target sequence occurs in vitro through a reaction catalyzed by a DNA polymerase in the presence of oligonucleotide primers and deoxyribonucleoside triphosphates in a defined reaction buffer. An important prerequisite for the amplification of the target sequence is that the reaction mixture contains no polymerase inhibitors. Amplification of the DNA is a cyclical process consisting of denaturation of the double-stranded DNA into single-stranded nucleic acid by means of heating, annealing of the primers to the target sequence at a suitable temperature, and extension of the primers, which are bound to both single strands, by a DNA polymerase suitable for PCR, at an appropriate temperature. Detection and confirmation of PCR products. PCR products are detected by gel electrophoresis or an appropriate alternative, if necessary, after isolation by means of a suitable separation procedure. The identity of any detected target sequence can be verified by an appropriate technique, for example by restriction enzyme analysis, by hybridization or by DNA sequence analysis. In the case of real-time PCR analysis, amplification and detection occur simultaneously. Aspects of PCR design. Size of PCR products. The size of the target sequence shall be selected to match the range of molecular mass available in the nucleic acid extract being analyzed. Example, for highly degraded DNA from processed foodstuffs, the size of the PCR product should ideally be in the range of 60 base pairs to 150 base pairs. For raw materials, a broader range of PCR products up to, for example, 250 base pairs is applicable. However, if prior experimental studies are carried out to determine the validity of primer sets yielding different-sized PCR products, these may be used on the matrix for which they have been validated. Primer design. The primer sequences should preferably have the following characteristics wherever practicable. Length of each primer, 18 to 30 nucleotides. Optimal annealing temperature 60 degrees Celsius, should be established experimentally, for example, estimated melting temperature less than or equal to 65 degrees Celsius. GC-80 ratio equals 50 if possible, or else as close to this ratio as possible. High internal stability. Avoid concentration of GS and CS in short segments of primers. Minimal 3' end complementarity to avoid primer-dimer formation. Minimal secondary structure. Minimal dimer formation with specific detection probes designed for the PCR. Software packages are available to help with primer design. PCR setup, detection and confirmation of PCR products. In the case of detection of the PCR products by gel electrophoresis, the size of the PCR products can be estimated using a suitable DNA size marker of known length to run in parallel with the PCR products under test. It may be desirable in some cases to confirm a positive or negative result for a particular genetic modification. This may be achieved by employing primers to an alternative target sequence. This is particularly suitable for confirmation of screening test results. A positive identification of the specific target DNA sequence may be confirmed by an appropriate method other than size determination of the PCR product, for example, by hybridization of the PCR product with specific probes, or by carrying out restriction analyses of the PCR product. The length of the resulting fragments has to correspond to the expected length of the target DNA sequence after restriction, or by sequencing of the PCR product, or other equivalent confirmation. If the primers used are designed to detect sequences derived from infectious organisms, a naturally occurring non-genetically modified organism such as a virus or a bacterium, then it is highly recommended that it be verified that the detected DNA is indeed derived from a GMO. This can be done by checking for the absence of other DNA derived from the infectious organism. Example, the 35S promoter is derived from cauliflower mosaic virus, and consequently detection of the CAMV35S promoter could be due to the presence of either GMO-derived and CAMV-derived DNA. By checking for presence of other CAMV-derived DNA, it may be possible to confirm the GMO origin of the CAMV35S promoter if no other CAMV-derived DNA is detected. Screening method for the detection of genetically modified plant DNA. CAMV35S promoter. This is a method for the detection of a variable copy number DNA sequence from the cauliflower mosaic virus, CAMV35S promoter. Due to the presence of CAMV35S promoter in many genetically modified plants, this method may be used to screen for the presence of GM plant-derived DNA. A false positive result can occur since the amplified sequence is derived from cauliflower mosaic virus infecting cauliflower and other members of the family Brassicaceae, cruciferae as well as Rhizidaceae and Solanaceae. Positive results derived from samples of Brassicaceae, Rhizidaceae and Solanaceae should therefore be treated carefully. Positive results may indicate the presence of a GM plant-derived product but should not be interpreted as proof of the presence of GM plant-derived products without additional confirmation. Principle: a 195 base pair DNA fragment from the CAMV35S promoter sequence is amplified by PCR and detected after separation by agarose gel electrophoresis. For identification of the PCR product, a verification step should be performed. Promoters are recognition or binding sequences for RNA polymerases, which are responsible for the expression of genes. The constitutive 35S promoter from CAMV is frequently used in genetically modified plants. Forward primer and reverse primer of CAMV35S promoter. Reaction mixes. The method is for a total volume of 25 microliter per PCR and the reaction setup is given in the table. All reagents shall be completely thawed at room temperature. Each reagent shall be carefully mixed and briefly centrifuged immediately before pipetting. A PCR reagent mixture is prepared to contain all components except for the sample DNA. The required total amount of the PCR reagent mixture prepared depends on the number of reactions to be performed, including at least one additional reaction as a pipetting reserve. Set up the PCR as follows. Mix the PCR reagent mixture, centrifuge briefly and pipette 24 microliter into each reaction vial. Add 1 microliter of each sample DNA or positive DNA target control or blank or other controls, mix and centrifuge briefly. The temperature time program as outlined in table was used in the validation study, general quality assurance and interpretation of the results. The target sequence is presumed to have been detected if the size of the PCR product corresponds to the expected length of the target DNA sequence, determined by comparison with products derived from certified reference material. The detection of fragments with the size of 195 base pairs indicates that the sample DNA solution contains amplifiable DNA of CAMV or GM origin within the assessed limitations of specificity. Screening method for the detection of genetically modified plant DNA, Agrobacterium tumefaciens NOS terminator. Method for the detection of a variable copy number DNA sequence from the Agrobacterium tumefaciens nuclein synthase, NOS, terminator. Due to the presence of the NOS terminator in many genetically modified plants, this method may be used to screen for the presence of GM plant-derived components. A false positive result can occur since the amplified sequence is derived from Agrobacterium, which is a soil bacterium present in nature. Positive results may indicate the presence of a GM plant-derived product but shall not be interpreted without additional confirmation. The potential contamination of the material with Agrobacterium or related bacteria should be considered. A 118 base pair DNA fragment from the NOS terminator sequence is amplified by PCR and detected by gel electrophoresis. The identity of the PCR product can be verified, for example by DNA sequencing. Forward primer and reverse primer for Agrobacterium tumefaciens NOS terminator. Reaction mixes. The method is for a total volume of 25 microliter per PCR and the reaction setup is given in the table. All reagents shall be completely thawed at room temperature. Each reagent shall be carefully mixed and briefly centrifuged immediately before pipetting. A PCR reagent mixture is prepared to contain all components except for the sample DNA. The required total amount of the PCR reagent mixture prepared depends on the number of reactions to be performed, including at least one additional reaction as a pipetting reserve. Set up the PCR as follows. Mix the PCR reagent mixture, centrifuge briefly and pipette 24 microliter into each reaction vial. Add 1 microliter of each sample DNA or positive DNA target control or blank or other controls, mix and centrifuge briefly. The temperature time program as outlined in table was used in the validation study, general quality assurance and interpretation of the results. The target sequence is presumed to have been detected if the size of the PCR product corresponds to the expected length of the target DNA sequence, determined by comparison with products derived from certified reference material containing the target sequence. The detection of fragments with a size of 118 base pair indicates that the sample DNA solution contains amplifiable DNA of nose terminator origin within the assessed limitations of specificity, interpretation and expression of results. Interpretation of controls. Each control has a valid value and, if the observed result for any control is different from the valid value, the analysis shall be repeated. The valid values for the controls are as follows. Positive extraction controls shall always be positive. Extraction blank controls shall always be negative. Positive DNA target controls shall always be positive. Negative DNA target controls shall always be negative. Amplification reagent controls shall always be negative. PCR inhibition controls shall not show significant inhibitory effects on the reaction. For qualitative analysis, the effect of inhibition may be less important than for quantitative analysis. Possible PCR results of the controls are listed in below table. These are used for interpreting and reporting the test sample result. All testing steps and results shall be recorded. Expression of a negative result. A negative result shall never be expressed as 0 or GMO not present. The following sentence shall appear in the test report. For target analyte X, GMO-derived material was not detected. Expression of a positive result. The following sentence or equivalent shall appear in the test report. For target analyte X, the presence of material derived from state-specific target sequence was detected. The identity of the GMO may be included, if known.